Hello and welcome back to Catapult Semi-Finals Biotech. We are going to now begin our second pitch session and we go directly to Paris, France, to meet Dr. Jean Garec with Biofta. Jean, over to you. Now, Jean, perhaps we need to unmute.
Okay, this is
Lovely.
very
Jean,
nice.
over to you, Jean.
Thank you.
Thank you.
So we use eye drops in the Romans 2000 years ago. And today, over 10% of the population use them every day and sometimes multiple times a day. It seems that the clicker doesn't work. The clicker doesn't work. Oh, I'm sorry. For tomorrow, pharma companies are developing intraocular injections. So it seems the future of ophthalmology. And despite all that, still millions of people lose sight because of disease for which there are efficient pharmaceutical treatments. And despite all that, still millions of people lose sight because of disease for which there are pharmaceutical treatments. I'm sorry, I'm completely lost. Because even the most efficient medications won't work if you don't take them regularly and if you don't respect the doctor's prescription. And this is the number one unmet medical need in ophthalmology, which is to guarantee a good compliance to treatment. I'll take an example, a patient with glaucoma. This person will need to use eye drops every day, 3 or 4 times a day, and this is forever since glaucoma is a chronic disease. Who can follow such a heavy treatment without missing any dose? And the direct consequence is that close to half of the patients don't respect their prescription, causing an irreversible loss of vision. So guess what? The current product development are in glaucoma to fight this compliance issue. Injections, again, but who would like to get that instead of eye drops? So there is a clear lack of innovation, and we confirm this major need with ophthalmologists from the US, Europe, and Asia. So what is our technology? It's a 2 millimeter mini tablet. We call it an ocular insert that is easily applied directly on the eye surface, exactly in the same way as you would do it with a simple contact lens. It's made of a new class of bioadhesive polymers that gellify and strongly adhere to the eye surface during 7 days for releasing a drug through an entire week for treating both front of the eye and back of the eye disease. We demonstrate that we have a sustained release and also that our insert is extremely well tolerated. So, of course, we are not the only ones willing to reduce the burden of ocular treatment for patients. And in general, we can observe 2 strategies that evolve in parallel. Either we try to improve the absorption of the drug at the eye, or we try to increase the duration of the treatment for a longer duration of application. So, and the technology that we have patented is the only one that is non-invasive, that offer 1 week of sustained delivery, and that increase the yield and efficacy of treatment. So, of course, looking at the market, global ophthalmic pharmaceuticals wait for approximately $30 billion. And we will at first focus on glaucoma. Why? Because we think this is where there is the greatest benefit for the patient, as it is a frequent disease with eye drops, etc. And our roadmap is then built initially on this first development program in glaucoma, and we anticipate revenues from a distribution license in 2025 after that we completed our phase 2 study. And we plan to leverage this data in order to sign co-development deals with additional companies putting their own drugs in our drug delivery technology platform. Finally, it's in the field of retina, where we expect the dramatic disruption on the market when we replace injections by a non-invasive and self-applied treatment. So we will initiate exploratory trials for diabetic macular edema in 2023. So then we already managed to secure financing until the first value creation milestone, and we target a 9 million euro in subsequent rounds to take us to the end of phase 2, where we could easily imagine an exit scenario. Finally, we are two co-founders with highly complementary profile, drug development for my partner and business in ophthalmology for myself. We completed our team with technical and scientific experts in ophthalmology, and of course, we have a strong medical advisory board.
Thank you, Jean. Thank you. Time is up and we have to be very strict with the timing. Let's now, thank you very much indeed for getting through your pitch, and well done. Let's now turn to the panel, the jury, and I can see we're already raising hands for questions. Renato, go ahead.
Yes, thank you. Thank you very much for the presentation. My question is about what is the final product? Is a combination product? Is a medical device? Because it's not clear from the presentation, and if so, what's the regulatory pathway? Thank you.
The regulatory pathway is a standard full drug dossier because we reformulate an existing drug within a new excipient. So we just designed a new pharmaceutical. However, due to the fact that the drugs that we're putting in the insert is not new, we can expect a 505(b)(2) procedures with the FDA. So it's a shorter pathway, but still a full drug dossier.
Thank you. I move to Julien, who would like to ask a question.
Yes, thank you. So on the product, who is positioning the product within the eye? Is it the patient? And what happens after the drug has been delivered? Is it solved by itself, or is there a procedure? Can you run us through that?
Yeah, excellent questions. So there are two things. The turnover, the polymers that we have covalently attached to the mucin at the surface of the eye. So that's how the insert can stay in place during 7 days. After 7 days, usually there is a turnover of the eye surface mucin. And once there is a turnover of the mucins, the insert just goes away and you find it in the morning in the corner of your eye. However, if you need to remove it early, or if the insert is still there at the end of the week, we have a solution, which is a kind of rescue solution, cysteine eye drops, very common, to remove the insert, and we tested, and that breaks all the link, and you can immediately remove the insert from the eye surface. So both solutions, either you wait the end of the week, or you need to remove it early, and you have a washing solution.
Alan, please, the floor is yours, and after, Emma.
Thank you for the presentation. A quick question, I guess, in terms of whether you're focusing on eye drop or injection. A number of the injectable applications are quite durable in terms of longer term. And if your, I guess, formulation provides once weekly, from what you're saying, will it really overturn, even though injections, nobody wants to do it, but if you have to have very frequent interventions with your technology, will it really overcome the longer term applications of injectables?
I return you the question. Did you ever receive an injection yourself in the eye?
No, not in the eye.
No,
but I guess in terms of
I will answer.
If it's 6 months or 1 year, that might be a challenge. That's the question.
No, I will answer. I will answer the question. First of all, it's very similar to eye drops. We interviewed, I think, close to 100 glaucoma patients, asking them proactively, okay, would you willing to change from your eye drops to this kind of device? You have to change to touch your eyes, etc. So there is no issue. Of course, we sign immediately if we can rid of the eye drops 3 times a day. So that was by far confirmed. 95% of the interviewed patients agreed to that. Second, I will tell you a little bit more about my personal story. I'm the son of 2 doctors, 2 ophthalmologists. My father used to do injections himself, and at the moment, reverse role. He is receiving injections as a patient. And believe me, 3 days before he's stressed, and it's painful 3 days after. So it's not something that is so common for the patients. That's not nothing to get an injection. And last of all, in the pandemic situation, you may be aware that many patients didn't go to their doctors. Why? Because they were afraid going to the hospital, and so many injection treatments have been delayed over time, and with, of course, loss of chance for the patient. So yes, I'm really convinced that we can overcome the injections because no one likes it.
Thank you. Emma, a quick question.
Very fast.
Quick
Yeah,
answer.
very fast. Yeah. So is there any influence in the absorption of your product depending on the level of hydration of the mucosa in the eye of the different glaucoma patients?
What will change is that the polymer enhance the penetration of the drug of the eye. There is a kind of reaction with the tight junction of the ocular surface epithelium, and then we are increasing the penetration of the drug inside. So we do not foresee any issue, except for very severe dry eye.
Thank you so much. Now, I'm so sorry to interrupt, but we have to now move on to our second startup in this second pitch session. We're leaving France to go to the Czech Republic. And we now meet Elżbieta Cardova of the startup Inantis. Elżbieta, over to you.
Thank you very much. Hello everyone, my name is Elżbieta Cardova and I'm a business development manager of a protein engineering company, Enantis. And I'm here to present the case of Enantis Pharma that's here to unlock therapeutic potential of Enantis engineered protein pipeline. We focus on novel protein therapeutics, specifically fibroblast growth factors, because these have a huge regenerative medicine potential. They're highly efficient, have minimal side effects, there's a shorter clinical development time, low immune response, but there's a huge problem, and that's their big instability. Luckily, Enantis has a solution for this, and that's the unique protein engineering approach that we use that allows for thermostability improvement just by substitution of a couple of amino acids in the protein. By this, we achieved significantly more stable molecules, up to 50 times sometime, with no stabilizing additives that allow for preparation of pharmaceutical formulations, and then the proteins have huge regenerative potential. As you can see in the video in the right corner, the native FGF2, for example, has a half-time of 9 hours, whereas the stabilized molecule, FGF2 STAB, developed by Enantis, has a half-life of more than 20 days. So that's a huge difference. We focus on growth factors in our pipeline. The main product for us is FGF2 STAB. It's a stabilized form of FGF2 protein, fibroblast growth factor 2, that's well known for cell proliferation and differentiation properties. It stimulates skin repair and regeneration, and it's a patented solution. We already have a proven track record on a research market because we have an exclusive supply agreement and multiple license agreements globally. But now we want to move towards the pharma applications for chronic wound healing. And that's specifically for chronic wounds such as diabetic foot and ulcers. We finished the formulation development, and now we have a preclinical safety design approved by Czech Medical Authority and started with the preclinical efficacy testing. And we have the whole pipeline of products. So apart from FGF2, there's also FGF10 STAB used for acute respiratory distress syndrome treatment that we also are co-developing. And then there are other molecules that are going to come into picture later on. The competitive landscape and market for FGF2 STAB are quite clear. When we compare the molecule with competing products, we can achieve less frequent administration and redressing. It's cost-effective due to a longer half-life. And we can achieve active wound regeneration. It's not just cleaning or debridement or supportive treatment. It's clear active wound regeneration because in the patients with diabetes, the healing process is impaired and we can kickstart the process again with our molecule. Here is the overview of the most close competing products. They're all based on some growth factors, but none of these are stable molecules. Some of them are even applied by injection, therefore it's painful, and the frequency of administration is way more often than in our case. We want to aim for once a week application or even less. And another thing is the distribution, which is not worldwide normally. So there's just one alternative treatment that's available for US and EU patients. And as you can see, 1 to 2% of the population of the US and Europe is affected by chronic wounds. So the chronic wound market is growing. And the attractivity of growth factors is also increasing in wound healing. We have a clear development plan. So first, we want to focus on FGF2 STAB for wound healing. In 2021, we already started preclinical efficacy testing. And in 2022, we want to start preclinical safety testing and then move towards phase 1 clinical. Similarly, with FGF10 STAB for lung regeneration, we want to follow in 2022 with formulation development, and then from 2023 to 2025, we would like to carry out the preclinical testing, both efficacy and safety. And then we have other FGFs in the pipeline for later. But for now, we would like to focus on the highlighted part with our Series A funding of €5 million that we're seeking for reinforcing the team of Enantis Pharma, GMP protein production, and hiring the CROs for clinical testing for FGF2 STAB and FGF10 STAB. We are ideally positioned for VC investment because there's a number of potential opportunities with partners for an exit. And we have a clear funding strategy. So we want to set up Enantis Pharma with all pharmaceutical assets, expertise from earlier phases, and we want to build upon the commercial success of Enantis with this Series A funding of €5 million.
Elzbieta, thank you very much indeed. We have to stop there, I'm afraid, because we have to respect our time.
Okay.
Yes, thank you. Well done. Let's now turn to our jury, who will also have 5 minutes to ask questions. Who would like to open the questions, please? With a show of hands. Lovely. Iris, I go to you.
Yeah. Hello. Thank you very much. Can you clarify? So your innovation is actually in the formulation, in the combination, when you combine it with other proteins that could be, for instance, growth factors, correct? So what is what
not
you have?
Really.
What is the core?
The core for us that we have a solution to stabilize the proteins, but we already started with fibroblast growth factors. So we have our own pipeline that we now want to commercialize in the medical field. And we want to start with FGF2 STAB for wound healing and followed by FGF10 STAB for acute respiratory distress syndrome. The platform is universal, but for now we want to focus on these two molecules.
Okay.
Yes, Lena, go ahead.
Changing the amino acid composition, even only if you change a couple of amino acids, you always risk that you always lose function as well, right? How many tests did you do already on that you were quite clear that the function is retained?
Yeah, we made plenty of tests. So actually the biological function is not influenced in any way. So the molecule acts as the normal native form. There's no difference. We've made plenty of tests in vivo, also in vitro, and now we want to move towards the preclinical tests that we already started with a small sample of animals, so we know it works. We just need enough number of animals for the statistics and so on. So we know it works. There's no problem with the alternative like functions or changed functions.
Thank you. Alan, I believe. Would you like to ask?
The presentation, very interesting. It's just a quick question in terms of the lung regeneration application. What specifically do you have in mind, and in terms of the delivery mode of action and formulation? How far advanced are you in that program? Thank you.
Yeah, we want to develop inhalation therapy, specifically by nebulization. So it's going to be targeted delivery into lungs, not a systemic delivery. So it's going to be less harmful to the patients. And we don't have the formulation yet, but we want to closely follow the Pulmozyme delivery system that's already marketed for cystic fibrosis. So the track record is already, well, the track is already proven, and we want to follow with our product. So it's going to be hopefully easy.
Would anybody else like to ask any questions?
You mentioned there's another FGF product under development, or maybe even already commercialized, this spray from a Japanese company. Could you maybe comment on whether you've done any sort of head-to-head comparisons? Do you have any sort of preclinical evidence demonstrating that increasing the half-life actually increases the efficacy?
Yeah, so we don't have the preclinical yet. We want to start with the preclinical thanks to this funding. But we've done some comparison with wild-type FGF2, which is the same one used in the spray. So the spray has really easy formulation. There's FGF2 wild-type plus some simple buffer. Therefore, we've already tested it a number of times in comparison with our product, and our product is way better. So we expect the same results in the preclinical.
And maybe to continue on that, which models would you consider appropriate here? Because people who have problems with open wounds often suffer from other indications. So how would you best model this preclinically?
Yeah, in preclinical, we're planning to use diabetic rats. So that has been recommended by our clinical authority here, the regulatory body. So that's what we're going to do in the first instance.
Okay, thank you.
We have 30 seconds left if somebody would like to ask a final question. Julian, over to you.
Maybe quickly, any other therapeutic areas that you want to look at and maybe partner?
Mm-hmm. Yeah, so apart from wound healing and ARDS, or the acute respiratory distress syndrome, we have other molecules for osteoarthritis treatment. So that's another one. And then there are molecules that are not developed yet that are going to be used in neurodegeneration and metabolic disease later on.
Okay, thank you very much indeed. Thank you for the pitch and for the great set of questions. We leave the Czech Republic to now go to Finland to meet our final pitch in this sequence, and we meet Pratik Singh from Fin Advance. Pratik, over to you.
Thank you. And hello everyone. I'm Pratik, founder and CEO of Fin Advance, where we are replicating human biology reliably. We are a team of engineers, scientists, and doctors based in Finland who are advised by a panel of industry-leading experts who have made successful exits in biotech and medtech industries to solve the problem of highly expensive and time-consuming drug discovery process, which you might know takes more than 10 years and upwards of $2 billion to get even a single drug to the market, which comes down to the basic problem of using extensive animal models in the whole R&D process. We end up developing drugs which are excellent for animals but fail more than 90% of times in the clinical trials. At FinAdvance, we are developing platforms where we are able to grow human cells, either normal or healthy, into tissues which would then replicate the disease. Our platforms keep the cells in a way as if they were growing inside the body. In this way, we are able to generate this universal platform where we are able to model neurological, respiratory, and even cancer disorders. Our customers have been able to use these platforms for preclinical research and replace as little as 24 animal experiments, and we are going towards replacing more than 200 comparable animal experiments on similar-sized platform. This preclinical R&D is a huge area, exceeding more than $150 billion. And out of this market area, we have been able to get some early commercial traction with some CROs and pharma companies, but we are capable of serving the whole market. In order to do that, we have a business model which relies on the hardware, the plate, combined with the consumables, the reagents, and high-value IP for high-value customers for replicating full disease models in-house. In 2 years of our existence, we have scaled quite rapidly having more than 800% year-over-year growth, both in revenue and customer acquisition. This gives us a very comfortable, I'm sorry for the graph, it has ruined up. This gives us a very comfortable and profitable path, hopefully going profitable in the next 4 years. All know-how is owned by the company with 4 filed patents and 3 more under filing this year. We do have a few competitors who are more research-based, and our biggest competitive advantage is the throughput. Even with the best competitor out there, we have 50 times higher throughput, and with the other ones, more than 200 times higher throughput. And we are industry automation compatible. All our platforms are handleable by industry robots. What started 2 years ago as a one-man company funded by EU today is 18% strong and is serving customers in more than 9 countries. Currently, I'm doing a late seed round of 4 million euros to increase our MRR to 300,000 euros with business development and pharma liaison offices deployment in US and Japan. Now, we are tackling a pretty big challenge and we have very big dreams. We believe that with our technology, we can help a big pharma company discover the first therapeutics for Parkinson's without using a single animal in the next 5 years. This is a pretty big challenge and quite bold, I must say. And that's why for these bold challenges, we are developing platform technologies. I finished a bit ahead of time, so I could take a few more questions than the normal. Thank you.
Thank you very much, Prateek. This is very, very interesting indeed. Let's open the floor to the panel, to the jury, for questions. And I see Renato would like to begin. Renato.
Yes, thank you very much for the presentation. I would like to understand more about the platform in terms of the final product. Is it considered as laboratory equipment, so no CE mark needed?
Exactly. It's a research use only. And we are going for ISO certification, but CE mark is not necessarily required.
Thank you.
Yes, Iris, and then Emma. Iris.
Yeah, so one question, how do you differentiate with regard to organo chips, 3D printed tissues and all these kind of things that are trying also to replace animal testing?
Really good question. We are an organ-on-chip technology, but most of the organ-on-chip companies have been focusing on replacing clinical trials. And the molecules what they get are tested on animals. We are replacing the preclinical trials. So the full chain is human only. And if we are working with animal pharma, then it's animal only. So our goal is to not only do the reduction in the clinical trial where you have to be involved with regulatory authorities like FDA and EMEA, but to go even much more earlier stage. Why not start with the in silico platform? That's our
Okay, so you are more into the preclinical part and the toxicology.
Exactly.
Okay.
Thank you. Emma.
You mentioned this organ chip technology, and you gave us this example of the Parkinson's disease on your slide. Are you focusing on this initial tracking on CNS or brain, let's say, assessing? What's your next challenge, or how do you plan to grow on that?
Definitely, Emma. One of our biggest application areas is overcoming the blood-brain barrier permeability. So we have a lot of drugs which show good efficacies on in vitro platforms, on 2D cultures, but it becomes a problem when you inject the drug and it's not able to cross the blood-brain barrier. We are able to do this exact same assay on our platform. So our customers have been replacing most of the animals in that specific area. So we are targeting a lot of neurological disorders using our platform. Thank you.
Who would like to, Iris, would you like to come back with a second question?
Yeah, you mentioned that you have some market. You mentioned that you have some market traction today. That you have 18 employees and are actually having some sales. And my question, very concrete, is what is your turnover right now? So just to see the potential.
Yeah, I'm quite happy about that because we are supposed to be pre-revenue. We are still under development, but the savings that our customers have made, they've been very happy to pay for the prototypes already. And this year we are turning 150,000 euros. So it will be, I think next February we will be 3 years old, and we were hoping to start making money after 4 years. I would
Thank
also
you.
quickly put, thanks. I would also quickly put, if there are no more questions or if the questions are coming, if you would know some good people who would be able to help me, please connect me with them.
Cillian, would you like to ask a question?
Yes, just a brief question. So you
Yeah.
mentioned the Parkinson's disease program. Can you maybe speak about the targets you're working on and how you select these, and maybe also walk us through how you plan to make the jump into clinical trials without the use of any animal models?
Thanks, Cecilian. Yeah, you might have heard that US Congress signed a bill this August on recognizing organ chips as an alternative to animal studies, and EMEA has a similar program. So their FDA has clearly recognized the advantages of these technologies. In terms of Alzheimer's and Parkinson's, or in CNS area, we have been developing the platform to study how novel therapeutics like peptides and antibody conjugates we can modify them in a way that they are non-toxic and still permeable across the blood-brain barrier. So that's the biggest area where the pharma customers are looking for a way to quickly assess which concentrations are non-toxic and have a bioavailability across the blood-brain barrier without using a single animal. We cannot do the whole clinical trial ourselves. Therefore, we would partner with some of these bigger organ-on-chip companies which have been focusing on the clinical trials only, closing the whole loop. Thank you.
Thank you. Thank you very much indeed. That brings our Q&A to a close. And likewise for our second session of pitches. Before we break, let's remember who we have met in the second part of the morning. We met Biotech in France, who are creating a best-in-class solution for patients suffering from chronic eye disease. From the Czech Republic, Inantis, presenting their unique engineering platform to substantially increase thermostability of FGF, fibroblast growth factors, for wounds and other indications. And finally, to complete in Finland, FinAdvance, providing a human-like biomimetic platform for drug R&D to reduce animal testing and decrease the total costs of drug discovery. What an exciting and inspiring and eye-opening morning. So please, let's now remember that we've got the networking platform where we can organise meetings to meet up with the startups, investors, delegates can get together. And of course, we are looking forward to getting your comments and photos on social media on the hashtag EITH Catapult. We are now taking a break for lunch, and this afternoon we will be continuing with our third and fourth pitch sessions, and of course with our expert insight, and we will be starting again promptly at 13:20 Central European Time, 13:20. So have a great lunch, great networking. See you at 13:20. Goodbye.